Good morning. Okay, so today we're going to start with a description of DNA structure, and most of what I'm going to say I'm sure you've heard already, but a little review wouldn't hurt. It's also important to understand DNA structure so that we can understand better how we are using that information to manipulate DNA molecules, putting them together and taking them apart. Okay, so the reminder here is the usual depiction of a double-stranded DNA. It's, of course, helical in 3 dimensions, and it has 2 strands, which are non-covalently linked to each other, but covalently linked within each strand. Each one is held together by the sugar-phosphate backbone, and the 2 strands are held together by a bunch of non-covalent interactions, which we will go through one at a time. Okay, so the phosphodiester bonds are covalent, but the hydrogen bonds, hydrophobic interactions, and base stacking are all non-covalent and can be disrupted by fairly mild chemical treatments. Okay, it's also important to know that the 2 strands are antiparallel to each other and what that means. That will be important, especially when you talk about DNA replication, etc. Okay, so what is a nucleotide? A nucleotide consists of a sugar, a base, and 1 or more phosphates. And it's good to get the vocabulary straight, even though we use these terms kind of loosely at times. The base, for example, might be adenine. Okay, so the adenine refers to the base, not the nucleotide. Although we usually just say adenine when we mean the nucleotide, just to confuse you further. But if I take my base and attach it to a sugar, that becomes a nucleoside with no phosphate. But if I add 1 or more phosphates to that, it becomes a nucleotide. So we have to, usually we're considering nucleotides, not nucleosides or free bases, but it's important to know the difference between those things. So now I'm going to take it apart and just talk about some of the properties of those different constituents of your nucleotide. Okay, starting with the bases, we've got 2 types, the purines and the pyrimidines. Purines are adenine and guanine. Pyrimidines are thymine, cytosine, and uracil. Now you know that uracil is the one that occurs in RNA, thymine occurs in DNA, and they base pair the same as each other, and the only difference between those molecules is this methyl group that's present on your thymine and not the uracil. Now, as molecules, as free bases, these are fairly hydrophobic because they don't dissolve very well in water. You can see they've got a fair amount of hydrophobic character because of all those carbon, and carbon hydrogen bonds. They're also heterocyclic in the sense that they contain both nitrogen and carbon as part of the ring structures. So that heterocyclic nature is characteristic of all of them, basically. They're also flat planar molecules, and so they actually physically stack one on top of one another like pennies when they're inside the double-stranded structure. Okay, so we're going to get to more of this later. I didn't mention, and should have, the conjugated double bonds in the bases are responsible for the high UV absorbance of these molecules. And this is very convenient because you can just use a spectrophotometer that has a UV light source to quantify the amount of DNA that's present in a sample, which is helpful. And the change in UV absorbance that occurs when the DNA strands are separated, something called the hyperchromic effect, which I'll get to in a bit, is also a convenient way of measuring whether the 2 strands are still together or whether they are physically separated from one another. Okay, so the sugar part might be ribose or deoxyribose. Ribose is used in RNA, and deoxyribose is used in DNA. Okay, so we're going to look at the sugar now, and it's important to know what the roles are of each of the 5 carbons in the ribo or deoxyribose sugar. Okay, so the first carbon, carbon number 1, is the one that attaches to the base. It's a carbon to nitrogen bond between the carbon in the sugar and one of the nitrogens in the ring structure, the purine or pyrimidine. The 2 carbon will tell you whether you've got a deoxyribo or a ribose sugar. It's shown here as an R group. Well, R, if it's OH, is a ribose. R, if it's H, is a deoxyribose. So the difference between the ribo and deoxyribose sugars is simply the presence of that hydroxyl group versus the single hydrogen in the form of the deoxyribose. Okay, the 3 carbon is the one that, if it's a linear molecule, would have the 3' OH sort of hanging out, would be the end of the molecule. But within the context of a polynucleotide structure, this 3' OH is responsible for forming the phosphodiester bond to the next nucleotide down in the line. And we'll see that better in a minute. The 4 carbon is the one that forms the ring with the 1, and the 5 carbon is the one that's sticking up out of the sugar and will be attached to the phosphates when we form the phosphonucleotide structure. And so that phosphate at the 5' carbon is the one that's going to attach to the next nucleotide before it as part of the phosphodiester backbone of the DNA molecule. Okay. The molecule shown is a ribonucleotide monophosphate, deoxyribonucleotide monophosphate. Okay, so the answer is B. You can tell that by looking at the sugar. The 2 carbon has an H on it, not an OH, and that means it's a deoxy. There's no oxygen there, and that says it's a deoxyribonucleotide monophosphate. And you can see it's a nucleotide because it has a sugar, a base, and a phosphate. So there's other kinds of questions that could be asked about this structure. Okay, the molecule shown contains A, purine or pyrimidine. Okay, so the answer is A, that's a purine. The purines have 2 rings and the pyrimidines have only 1. Okay. So now we want to talk about DNA, single strands, before we put the molecule together, right? So single strands of DNA or RNA consists of deoxyribo or ribonucleotides that are linked together by phosphodiester bonds. And so it's important to realize that it's not the bases themselves that are involved in the phosphodiester bonds. They're just sort of hanging out there into space. It's the sugars and the phosphates that are linking the adjacent nucleotides. Okay, so also important concept is that single strands have polarity, which is to say direction. And we can, by convention, write the sequence of the bases in the 5' to 3' direction on the molecule. Okay, so the single-stranded thing, you can easily think about it as, or I think about it, is that each sugar has itself a directionality or a polarity. I think of the top of the sugar being the 5' sticking out carbon, and the bottom of the sugar being the 2 and 3 carbons that are sitting flat on the base. So the sugar itself has direction to it if you consider the 5 carbon to be the top and the 2 and 3 carbons to be the bottom. Each sugar has itself a directionality, which when you link them together imparts directionality to the overall molecule. So polarity refers to direction. It doesn't refer to charge or chemical polarity. All right, so when I represent this, I can think of, here's my sugar, my 5 carbons sticking up, my 2 and 3 are sitting there on the bottom, that this molecule has a direction, 5 to 3, with the 5 sticking up on the top, each one. So when we put those together, putting the little phosphate linkers in between the sugars, we have the top sugar, 5 to 3. The next one itself is the same direction, 5 to 3, 5 to 3, 5 to 3. So when we link those all together, the overall direction has this one 5 at the first, running down to the bottom 3. So the individual sugars impart the direction to the overall molecule, which is called polarity. Okay, so when you put 2 strands of DNA together, you realize that the 2 strands are actually antiparallel, which is to say that the sugar-phosphate backbones run in opposite direction. And that's important. They're also complementary, and I'll get to that in a sec. Okay, so if you look at your double-stranded structure, you can see you've got your 5 carbons sticking up here. This strand is running 5 to 3, top to bottom as drawn. And the last one is at the bottom. That's the 3' end of the molecule. So you can follow the sugars 5 to 3, 5 to 3, 5 to 3, etc. Now, if you look at the opposite strand, the other strand, you can see that the phosphodiester sugar backbone is running in the opposite direction. On that strand, your 5 is at the bottom, and that the 3 is at the top. So these sugars are literally upside down relative to the other strand. So this strand is going 5 to 3, 5 to 3, bottom to top, and the one on the left is running 5 to 3, 5 to 3, top to bottom. So that's what anti-parallel means. They really are running in opposite directions. Okay. So when you get to your double-stranded structure, you have to consider the forces that are holding it together and the forces that are driving it apart. Now, one thing I should have said earlier about the phosphates, each of those phosphates, which is used to link the adjacent sugars, has at least one negative charge by means of this ionized oxygen. So the two strands actually tend to repel each other because you've got a negative here and a negative on the other side. So charge repulsion is something that needs to be overcome when you're going to try to put DNA molecules together. So that drives the two halves apart. Okay, the hydrogen bonds are interactions between the actual bases, which are sticking into the center of the helical structure, actually. And you can see that if you look at this GC base pair, there are hydrogens, 3 of them, that are being shared between the guanine and the cytosine nucleotides. Right, so those hydrogens are interacting with the ring structures of both of the nucleotides. Now, each hydrogen bond is relatively weak, but if you have gazillions of them all on the molecule, it ends up being fairly strong. Notice, too, this is a GC base pair. It has 3 hydrogen bonds. The next one's an AT base pair, and that has only 2 hydrogen bonds. So the GC base pair is stronger than the AT base pair because it has 3 hydrogen bonds instead of only 2. So the complementarity idea, of course, is that when you have a G on one strand, you have a C on the other, and vice versa. Whenever you have a T, you have an A, etc. So you do get the same base pairings throughout. All right, so we mentioned hydrogen bonding between the bases, the GC base pair being stronger than the AT. We need to mention, and we also mentioned the charge repulsion. We need to now mention a little bit about base stacking, which stabilizes the double-stranded structure. Okay, so the traditional, we've got our hydrogen bonds in here. You can see this GC base pair has 3 of them, etc. But realize within the molecule, those flat penny-like molecules are sitting one on top of one another. So they're actually interacting with each other within the molecule itself as a result of interaction between the pi orbitals on the 2 different purines and pyrimidines. So that tends to stabilize the structure because you have interaction not just across the molecule by means of the hydrogen bonds, but also the stacking of the bases themselves. Okay. Which of the following is most directly responsible for the high UV absorbance of DNA molecules? Double bonds in the base ring structures, double-stranded character of the molecule, phosphates and the resulting negative charge, the deoxyribose sugars, hydrogen bonds between the bases on both strands. Okay, so the answer is A, of course. It's the double bonds that are absorbing or leading to the absorption of ultraviolet light. Which of the following statements about bases in double-stranded DNA is false? They are involved in hydrogen bonding to the opposite strand. They contain nitrogen and carbon ring atoms. They may be purines or pyrimidines. They are covalently bound to sugars. They are very soluble in water. Okay, all those statements are true except the last one. They're fairly hydrophobic molecules and they don't like water at all. Okay, so now we're going to talk about, now that we have built our double-stranded DNA molecule, it's going to be very important in the future, not so much to denature DNA, but to hybridize it, hybridize 2 different DNAs to each other. So important concept, of course, is that the 2 strands are held together by non-covalent bonds, and so they can be separated by fairly mild treatments. Okay, but other factors are going to come into play, namely the chemical environment and the base composition of the DNA itself. Okay, so here's what's called the DNA melting curve. Ignore the y-axis for a moment. We can consider this as being percent single-stranded, or double-stranded rather, and sees what happens to the molecule as we increase the temperature. So if we start off at room temperature and gradually raise the temperature, there's going to come a point where the strands begin to separate from one another because those non-covalent bonds are being broken. And there's a temperature called the melting temperature, Tm, at which that reaction is basically 50%. Eventually, as you keep raising the temperature, the molecules separate completely so that they're now physically separate from one from the other. Okay, so if you look at the axis on the previous slide, it doesn't say percent single-stranded like I said. What it's actually looking at is UV absorbance of the molecule. Okay, this is the hyperchromic effect, and it's used to see that the DNA is actually melted. Okay, so the phenomena here is that if you melt or denature the double-strand DNA to form single strands, the UV absorbance increases, which is called hyperchromicity. Okay, and so what happens to the molecule as it starts to melt is you lose base stacking and you also lose hydrogen bonding as the thing dissociates. Okay, so the unstacked bases evidently absorb UV more strongly than the stacked ones in the double-stranded structure, so that when the molecule starts to separate, the UV absorbance actually goes up as a result of the loss of those interactions among the bases. So this is useful and it allows the state of the DNA to be observed. The change in absorbance lets us observe the melting. But you could also just think about it as percent single-stranded, which is what I do. Okay, so here's another picture showing the influence of GC content, because the melting temperature, or hybridization temperature, if you're putting things together, will depend to some extent at least on how many GC versus AT base pairs you have. So this is a melting curve, as before, where we started off with double-stranded DNA, gradually raise the temperature, and then we get to the transition, the midpoint of the transition, and then finally the two strands are completely separated when the temperature is raised all the way to 85 degrees. So that's a standard melting curve. Okay, the other figure on this slide shows the influence of GC content, right? So what we're looking at is the melting temperature of a variety of molecules as it relates to the percent of GC within the molecule. So the very, very low GC content will melt at, let's say, 80 degrees. But if the GC content goes higher, the melting temperature goes higher, which makes sense because the hydrogen bonding is stronger if you've got more GC base pairs. Okay. So that's all we need, actually. Okay, I didn't do this one in class, but we can do it anyway. The genome of which organism, A, B, or C, has the highest proportion of GC base pairs? A, B, or C? Okay, so the answer is C, because it has the highest melting temperature. It takes a higher temperature to get your strand separation. So this is going to become important when we try to anneal DNA molecules together, put them together for various purposes. So it's important to know something about the influence of the chemical environment on the DNA molecule itself in order to try to do experiments like that. So clearly, high temperature will favor strand separation, also known as dissociation, denaturation, melting, because the non-covalent bonds can be broken at high temperature. Now, the chemical environment is also extremely important. One important feature is the effect of salt on whether the DNA molecule is going to melt or not. So high salt actually favors the strands staying together, right? As a result of this, it actually raises the melting temperature of a DNA. Okay, the reason for this is that the salt neutralizes the negative charge on the phosphates, and that thereby removes one force that's driving the strands apart. So salt tends to allow the DNA to stay together at a higher temperature than it would otherwise with no salt, because it has neutralized the negative charges. There's also effects of molecules added to the solvent, which actually will favor dissociation or melting, and they do that by competing for hydrogen bonds. Okay, so the examples that we normally use are formamide and urea. So let's look at the effects of salt first. Salt neutralizes the charge repulsion. So in the graph on the left, we're looking at a series of DNA molecules. Each point is a DNA molecule, which differ from the others in their GC content. And we're going to look at the effect of salt on that reaction. So this graph is a lot like the other one where you could see the higher the GC, the higher the melting temperature. The melting temperature is given on the y-axis. So this gives us the curve showing the effect of GC content on the melt, which is just what we saw before. Now this graph, the line on the left, was done in the presence of 150 millimolar sodium chloride. The lower line, however, is the same series of molecules, but instead of having 150 millimolar sodium chloride, you have 10 millimolar sodium chloride, or sodium phosphate, same idea. So there's a definite shift to lower temperature of melt because you don't have the sodium chloride there in order to neutralize the negative charges. So we have a shift from, average melt temperature being, let's say, 85, down to an average melt of being, say, 70, as a result of the influence of salt. And the graph on the right basically illustrates that general idea that if the salt concentration is lower, the melting temperature will be lower. If the salt concentration is higher, the melting temperature will be higher. So this just says what I just said, I think. Okay, so molecules that might be there in the solvent that destabilize double-stranded DNA will cause it to melt at a lower temperature. And that can be convenient for experimental purposes, actually. And you see the use of formamide and urea really quite a lot. Okay, so what they do is they form hydrogen bonds with the bases, and this competes with the real hydrogen bonds. So you can imagine the molecule sort of melting a little or peeling apart, then the formamide or urea in the solvent will be able to interact with the bases and prevent the base pairs from reforming. So formamide, for example, has the ability to interact with adenine by sharing hydrogen with it, and with cytidine, cytosine, etc., as does urea. So what they are doing is creating unnatural or not desired base pairs, really, and replacing those unnatural base pairs with the real ones in the molecule. So that clearly will favor dissociation or will cause the molecule to fall apart more readily because you've lost some of your base pair interactions that are important for keeping it together. Okay, so we're saving this for next time because the more useful idea about DNA molecules when you're trying to melt them is what you're more often trying to do is hybridize them, which is to take two different molecules and put them together and have them form a double-stranded structure from the single strands. So this is a very important process for many things, including sequence and PCR, etc. Okay, so we need to consider what the chemical conditions are like in order to derive conditions where it's good, we can get the good, correct molecules that we're trying to form in a test tube. So I'll save the rest of this for next time.